Hello everyone, I'm a PhD student at the Institute of Science of Evolution of Montpellier in France, and my thesis is on the adaptation of Paphionidae to their host plants and their diversification. My thesis project involves studying the hypothesis that the diversification of phytophagous insects is directly linked to that of plants via an arms race. It was Ehrlich and Raven who introduced the concept of co-evolution in 1964, using the interactions between butterflies and angiosperm to try to explain the impressive biological diversity of these groups. The main principle of Ehrlich and Raven's theory is that the evolution of the plant chemical defenses is closely followed by the biochemical adaptation of insect herbivore, and that newly evolved detoxification mechanism lead to an adaptative reduction of insect herbivorous lineages due to newly available ecological niches. The idea is to carry out a genomic study of molecular evolution in Paphionidae in order to reveal the genomic consequences of host plant changes in relation to conservatism. We would like to know if there are adaptative signatures in the genome caused by host plant changes for the different groups of Paphionidae. And if what are the genomic changes have evolved in response, and if there are the same genomic changes behind all host plants, or if they differ between host plant families. This project involves replicating a study carried out by Rémi Alliot in 2021, who observed adaptation signature in the genomes of Paphionidae at the genus level, by finding a significantly greater percentage of genes under positive selection in branches where a change of host plants had occurred, compared with branches with no apparent change. In the studies, I lacked in confidence in the gene sample, particularly at the ontology level. So my work began with the creation of a richer and more reliable database of orthologous genes. To do this, I used the complete genomes of 30 species that provide a good representation of the Paphionidae family, since they cover all existing tribe and 17 different genera out of the 27 that exist. On these genomes, I carried out the first annotation step using 3 different programs. Busco, which uses a library of 5286 Lepidoptera genes, and Miniprot and Scipio, to which I supplied as reference the complete genome sequence, the complete protein sequences of these three different species in red. The second step was to retrieve as many orthologous genes as possible from among genes annotated by the 3 programs, but independently. To do this, we use the AutoFinder program, and we select a single copy orthologous genes when they are charged by at least 5 species, and also if they had 1 paralog, but only when the paralog was a result of a recent duplication. This step enabled us to recover a number of orthologous genes from among genes annotated by Busco, Miniprot, and Scipio. Then we had to align them and clean them, which reduced the number of orthologous genes for each. Here it's a Venn diagram of the number of orthologous genes found, we can see that the three different methods have in common 75 orthologous genes, and that each method has found between 1,000 and 2,000 orthologous genes of its own. If we add up all the parts of this, we end up with 9681 different alignments to build our Papilionidae orthologous genes library. We then created the alignment profiles, and this now constitutes our new library. Thanks to this new database, we set once again to study the presence or the absence of significative signature in Papilionidae. Yes. You choose to start with the Leptocercini tribe, relying on the phylogenetic reconstruction from Elliott's thesis, which provides inferences of host plant regime at family level, as well as geographical area for around 150 species, which represents almost all existing species for this tribe. It required more than recovering orthologous genes from the complete genomes of the available species. And to achieve this, we use the same annotation methods. I use the example of species annotation to show that each program can annotate a very different number of genes. Then I search for the orthologous gene of each Leptocercini species on the basis of our Papilionidae orthologous genes library. To do this, we use several programs which search for reciprocal correspondence between the sequences of the annotated genes of the Leptocercini species and the profiles of the orthologous genes of our library. Finally, to align the orthologous genes of the Leptocercini species, we add them to the alignments of our library to which they correspond. And this and the last step of cleaning. And this finally gave us 9657 alignments for the Leptocercini, on which we are currently studying the presence of or the absence of adaptative signatures. As was the case for Alieu's study, we thought to define the states of change of the branches to constitute the sets on which to determine selection pressure. We use the phylogeny reconstruction showing host plant regime inferences to define branches where change did or did not occur. And we followed the same approach on the phylogeny showing geographic areas. And in the set of branches, we selected none had a radical change of host plant regime or geographic area. In total, we categorize 21 branches with no change, 28 with a change of host plant, 22 with a change of geographical area, and finally 14 on which both types of change had occurred. This phylogeny of the 150 species of Leptocercini shows the branches selected and the category assigned to each. Unlike Alieu's study, we Yes, unlike earlier study, we began this study by selecting only terminal branches at the species level and not at the genus level. For selection pressure inferences, we used iFi software and its RBSReal method, and we run the analysis on the 85 branches selected and all the orthologous gene alignments for which at least one branches was represented. Here it's 9350. And the minimum coverage for a branch was 3266, and the maximum was 7325. And of the 9350 used, 6668 were found to be significant to positive selection. So, our results were to, our first results were to investigate if we were also detecting adaptative signature in lineages, in lineages that had the change host plant regime. Just finally, as the earlier study had done, and for our part, we found no difference in the percentage of gene under positive selection between our category of change. But my study is still at a preliminary stage. But we can already raise a few points for discussion, and notably on the differences between our current analysis of and that of earlier study. Thank you for your attention.